Well, good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Every Patient Matters, Bespoke Digital Endpoint Strategies for Rare Disease Drug Development. My name is Ryan Muse and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and your comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit your questions as well via the comments tab. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming to those who have registered on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario is a leading provider of endpoint data solutions to the clinical trials industry, generating high-quality clinical evidence for life sciences companies. They offer comprehensive evidence generation solutions that combine medical imaging, eCOA, precision motion, cardiac, and respiratory endpoints. Now I'd like to introduce our speakers for today's event. Dr. Mark Oppler is the Chief Research Officer of eCOA Neuroscience at Clario. In this role, he directs scientific research and leads ongoing studies in psychiatry, measurement development, psychometric validation, and other areas of neuroscience. He is also the Executive Director of the PANS Institute. His academic research focuses on etiology, phenomenology, and treatment of serious and persistent mental disorders. Dr. Scott Hunter is a senior scientific expert specializing in neurodevelopmental and rare diseases, as well as epilepsy. With a distinguished background as a former professor of psychiatry and behavioral neuroscience and pediatrics at the University of Chicago, Dr. Hunter is a recognized authority in the field. His expertise spans the design and validation of clinical outcomes assessments and clinical trial endpoints identification. Dr. Thomas First is the Chief Science Officer at Clario, where he leads scientific strategy and innovation across imaging and endpoint technologies for clinical trials. He has extensive expertise in advanced imaging modalities, including XR, CT, and HRCT, MRI, ultrasound, DXA, QCT, and photography. Dr. First has been at the forefront of developing and applying novel methods to generate high-quality, reliable data for global studies. And Dr. Vic Patel is a board certified in cardiology and holds a PhD in biophysics, having conducted extensive research on the mechanisms of cardiac arrhythmias. He has a faculty or is a faculty member at the University of Pennsylvania School of Medicine for 11 years, where he directed molecular arrhythmia research and practiced clinical electrophysiology. Dr. Patel then transitioned into drug development and cardiac safety. But without further ado, I'd like to go ahead and hand the mic over to our first speaker today, Dr. Mark Oppler. You may begin when you're ready.
Thank you so much, Ryan, for the enthusiastic introduction. Much appreciated. We can go to the first slide. Just to run through today's agenda, we are going to be covering a range of topics under the umbrella of rare disease trials, from unique challenges and imaging endpoints to digital endpoint strategies and cardiac assessments in rare disease. We're going to finish today with some wrap up and Q&A, going over the questions that you, the audience, are kind enough to submit. Let's go to the next slide, Ryan. So some of you are kind enough to participate in our poll that we put out earlier. I think the results are very interesting. We asked you all to tell us what you thought the biggest challenge in rare disease clinical trials was. Was it awareness or engagement, the regulatory hurdles, the complex measures and endpoints, or the small patient populations? And by a reasonable majority, most of you said it was small patient populations, although obviously a fair number of you also felt that some of the other challenges represented somewhat more of a hurdle. So thank you for participating in that. We are going to be addressing all of these topics broadly as we go through today's presentations, and look forward to seeing how that evolves as we go through the next hour. Moving on to the next slide, I just want to begin by making a brief observation about in my worldview, why it is that rare disease studies are often so challenging for those of us working in drug development. I'm often reminded of the story of the Jamestown colony. At the beginning of the 17th century, the Virginia Company in Virginia Company in London, founded one of the early British settlements in Jamestown, Virginia. And the assumption was, as they were getting ready to embark on this venture, that they were mostly going to be doing things like mining gold, obtaining rare silks, creating valuable perfumes, and cutting precious gems for jewels, and shipping all of these luxury goods to Europe. So among the settlers who showed up on that first boat at the Jamestown settlement, we saw 2 goldsmiths, 6 tailors, a jeweler, and a perfumer. Now, knowing what we know today, and even what we can reflect on with our history about the conditions that they actually found in the swamps of Virginia, those 2 goldsmiths, tailors, jeweler, and perfumer, they all fared about as well as you could expect. The stories of the early experience in Jamestown were rife with all kinds of death, disease, and tragedy. While nothing so drastic often happens in our rare disease studies, when we show up to do these studies, going on assumptions that they're going to go a certain way, and that all of the conditions and strategies and approaches that we're used to in the conduct of routine clinical trials, if there is such a thing, of course, that all of that knowledge or standard operating procedures and practices all will apply perfectly to rare disease trials. Well, that ordinarily goes about as well as you'd expect. If we go to the next slide, let's look at a different rubric for this. I believe also that when we move from early to later phase in drug development, especially in rare disease, there's often something that's lost. And I think many of us on the call today, both panelists and participants alike, can probably tell any number of war stories about promising treatments that didn't pan out. And among the common themes that I hear when I talk to folks, I hear about all kinds of great preclinical data on promising molecules with strong links to the pathophysiology that we believe underlies many of these rare diseases. Unfortunately, in our effort to translate that into later phases of development, we're often hampered by things like short treatment regimens, very small samples, topic we'll talk about throughout today's session. And of course, the challenges of proving in a rigorous way that often are subjective outcomes, whether they're patient-reported, caregiver-reported, or something similar, that these treatments actually work. So how do we move beyond both of these broad conceptual problems? How do we move beyond, first, the limitations of our own expectations and standards, and also the challenges of moving from promise to practice in the study of rare disease? Let's go to the next slide. I believe that there are many things that we can do, many of them common sense, some of them take a little bit more finesse, whether it's optimizing the treatment regimen to make sure that it actually works, meeting the needs of the population, not just, of course, participants and caregivers, but also the investigators and the research sites. But most of all, and I think one of the things we're going to talk about a lot today, are the need for adaptive, flexible methods, tools that are actually appropriate for conditions on the ground, as it were. And as we walk through today's sessions and the presentations by my colleagues, they're going to share their expertise with you on this very topic. And we'll see how in each of these different facets, from cardiac to imaging to the use of digital endpoints, adaptivity and flexibility can succeed often where our prior assumptions have failed. With that, why don't we go to the next slide? And I'm very pleased to introduce Dr. Thomas First, Chief Science Officer for Medical Imaging at Clario, who will be discussing considerations for imaging endpoints in rare disease trials. Thomas, over to you.
Thank you, Mark. We can go to the next slide. As Mark indicated, there are some special challenges that we face in rare disease trials, but we also face many of the same challenges that we would in any other indication. But those challenges are heightened by the scarcity of patients and our desire to maximize the data that we can collect from the rare disease patient, but also some of the challenges with regard to the health and fragility of this patient population. Today, I'll be talking about some things that are very common across all types of trials, selection of endpoints, designing a protocol for collecting those endpoints in a multi-center setting, finding study participants, which is always a challenge in rare disease, and connecting those participants with imaging devices, and then the importance of collecting high-quality images and conducting precise analyses. For our presentation as a whole today, we're going to use Gaucher's disease as a case study. It's a common thread throughout all the presentations today. And on the next slide, we'll just introduce this for those who are not familiar. Gaucher disease is a rare inherited metabolic disorder caused by insufficient glucose cerebrosidase activity, leading to harmful lipid accumulation in cells. As such, it's a member of a larger family of lipid storage disorders. Signs and symptoms of Gaucher disease vary widely. They include skeletal disorders, enlarged spleen and liver malfunction, anemia and low platelet counts, and neurological and cardiac problems. On the next slide, you can see this creates a rather rich environment for us to deploy imaging endpoints to collect information to support safety and efficacy of new drugs being tested. With imaging, we can use MRI or CT to look at liver volume and spleen volume. MRI can also be used for brain imaging to look at structural changes or connectivity, the latter being done using functional MRI techniques. Bone issues can also be evaluated with MRI as we look at bone marrow abnormalities, which are evaluated using a visual scoring system called bone marrow burden, a specialized assessment for Gaucher disease. It also allows us to look at lytic lesions and fractures, and in some of these trials, bone mineral density is also evaluated with imaging techniques. And finally, we can use cardiac MRI or echocardiography to look at cardiac structure and function and valvular calcifications. The next slide, I wanted to get into the topic of selecting imaging endpoints. Now, regulatory guidance is available for some rare diseases, but not all rare diseases. And as such, you find yourselves breaking new ground. My experience, though, is that regulators are very eager to collaborate on trial design and very welcoming of your ideas for new and innovative endpoints that might be useful. Those endpoints, as a consequence, might be novel and untested. Still, they have a role in trials as secondary and exploratory endpoints. And again, in my experience, especially in rare diseases where we're dealing with very small patient populations, I find that these additional endpoints contribute to the totality of evidence supporting the efficacy and safety of new drugs and contribute heavily to the evaluation of these therapies. Those endpoints might be complicated to acquire and analyze, which certainly introduces some challenges for the real world setting of a multi-center clinical trial. So control and standardization of the imaging is important. And lastly, rare disease patients are very precious, and we find ourselves often faced with this problem of we have so few patients, we have so much imaging that we'd like to do and so little time to get it done. I think it's important that we avoid the trap of trying to do too much. And I have seen some trials that are designed with very, very extensive imaging protocols that require long imaging times, multiple modalities, multiple imaging sessions, or perhaps imaging has to be spread out over 2 days. And I think it's important to remember the burden on the patient and to choose wisely which modalities and which endpoints you want to include. And that might mean letting some things go. On the next slide, now that we selected our endpoints, we have to design an imaging protocol that can be used to acquire images reliably that support those endpoints. And some of the factors we need to consider include the radiation dose. X-ray, CT, and PET-CT all involve exposure to ionizing radiation, and that may limit which modality we use or how frequently we can employ that. Choice of modality also has to consider factors like convenience and cost. For example, liver and spleen volume can be measured quite accurately and precisely with MRI. That's rather costly. Ultrasound is an alternative that can provide approximate measures of volume. It would be perhaps more convenient and less costly. Also need to consider whether special equipment is needed and whether that might put limitations on which imaging facilities or which investigator sites or which regions of the world you might be able to recruit patients. Frequency of imaging is important. It impacts patient burden. And in addition, we have to keep in mind any limitations that come from the patient population that we're trying to study. For example, are there contraindications like implanted devices that would prevent use of MRI imaging, or poor renal function that prohibits the use of contrast media in this patient population, or other patient restrictions? Are they able to hold their breath to avoid motion artifacts during imaging? Is body size or positional limitations going to interfere with the acquisition technique? These are all important factors that will influence which modality and which endpoints one will evaluate. Moving on. After we've set up our imaging protocol, we have to roll this out into the real world, and we want to first and foremost find patients, and that means going to the best recruiters. But the best recruiters are not always the best imagers. So it's always important to partner with an imaging core lab that has a proven ability to qualify and train novice sites on advanced imaging techniques. One possible solution to both of these problems is academic centers of excellence. It provides a nice nexus of patient access as well as advanced imaging. These referral sites may be supporting a large geographic region, and they typically have access to advanced imaging techniques and experience with application of those in the patient population of interest. The drawback there is that they may be involved in many clinical trials, and as a result will not be able to contribute as many patients to your trial as you would like. We could go back to the last slide just for one last point there. Competition for patients might lead us to go into new geographic regions and cast a broader net, which reinforces those challenges of finding the right imaging capabilities. And then the imaging referral site is an option where one single imaging site that has advanced capabilities supports multiple nearby investigator sites. Okay, we can go to the next slide at this point. Rolling things out into the real world, we want to ensure we collect high quality images that are supporting the analysis endpoints. Site imaging personnel need to be trained. There's a variety of ways in which that can be done. Because of the rarity of patients, it's often valuable to acquire some test data to iron out any issues before the first patient visit. That may be a phantom test data or human volunteer. Image QC is important to ensure the images are fit for purpose, and that means from time to time images or exams may be rejected and need to be repeated. That can raise some challenges because participants might travel a long way to the clinic, and coming back for a repeat exam can be a challenge. Working with SMEs and KOLs is also advantageous at this point, either as a reader or sometimes if the KOL's schedules are too busy as an advisor. And making use of computer automation is helpful, but remember that those tools might have been developed for a different population, maybe an adult population, and may not work as well or effectively in a pediatric population. Next slide. Many of the rare diseases we encounter have genetic origins and as a result involve pediatric populations. And there's some special considerations for the pediatric population I wanted to reference. First is radiation avoidance. Obviously, it's a concern for all patients, but heightened in the younger patients. So decisions about where MRI or ultrasound could be used to avoid ionizing radiation will be made. Participant compliance is important. Utilizing child-friendly environments, shorter scan times, or sedation alternatives to avoid motion artifacts are all important considerations. And we have to remember also in the pediatric population, the patients are maturing and growing and developing, and interpretation of data may be a challenge. It's always helpful when available if you have normal reference data from a healthy population that you can compare results to, that will allow more accurate interpretation of the data. So identifying and locating that reference data is important. Go to the next slide. One of the other factors related to pediatric rare disease is bone safety. A lot of pediatric rare disease conditions have direct effects on the bone, like osteogenesis imperfecta, achondroplasia, hypophosphatasia. But other conditions may not directly affect the bone, but there are regulatory concerns about impacts of new therapies on bone metabolism, skeletal maturation, achieving peak bone mineral density, and peak height. So there's a lot of interest to evaluate bone endpoints in pediatric clinical trials, whether that indication is directly affecting the bone or not. So assessments of bone age, growth plate health, and bone mineral density are often included in these trials. That was the last of the slides that I had to present today, and we'll turn things over to Scott Hunter, who will take us through the challenges in ECOA.
Thank you so much. It's a pleasure to be with everyone today and to follow such important information. When we're doing a clinical trial, we're also looking at, beyond imaging, other ways in which we can obtain data, often data that is tied to scales and test batteries that allow us to understand functioning in the patient groups that we're going to be seeing. And it's particularly of importance with regard to rare disease groups, where we may be looking at not only cognitive development, but also behavioral development simultaneously, and needing to have a range of tools that would allow us to actually assess that information. Next slide, please. When assessing in clinical trials, we're really focusing a lot on the utilization of a number of different self-report, caregiver report, and the clinician observation measures, in addition to performance measures that allow us to understand how functioning over time is developing, or how functioning may be impacted and changing when talking about a group across the lifespan. These measures are particularly vulnerable to a variety of administration and completion errors by trial raters, and some of them are often fairly complex in terms of the processes for both obtaining and recording the data and then scoring it. So even with administration experience and trial-specific training that is often very much a part of the clinical trial process, these errors can occur, and we often see them most when paper-scale administrations take place. So with the advancement of electronic data capture, we've actually been able to move into thinking more effectively about ways to better support the obtainment of reliable and valid data. Next slide, please. The rationale that comes about for using ECOA, the electronic capture of observer and clinician data, for the patient-reported outcome measures, the clinician-reported outcome measures, observer-reported outcome measures, and the performance outcome measures, or PROs, ClinRos, OBSROs, and PERFOs, as they're well noted in the field, that gives us a chance to be able to utilize tablets and desktop computers as a means of more efficaciously and efficiently obtaining these assessments, and also being able to address the complex scoring components that often go along with them. Utilization of ECOA allows the examiner, both the investigator and the rater, to focus directly on correctly and efficiently obtaining data across all forms of clinical assessment administration. With regard to putting these scales into ECOA, we're able to actually build important algorithms directly into the system that allow us to facilitate, guide, and intervene as necessary to ensure we have a valid administration as well as the scoring of this data so that it can actually then be utilized very quickly and effectively by the clinical trial sponsor. ECOA also fosters one of the most direct means for allowing required regulatory checks on validity, administration, efficacy. One of the things that has become very important in clinical trials now is how we address and meet the demands of the regulators to ensure that we have the most appropriate sources of data and the appropriate range of scores. Particularly important in rare disease, and in the pediatric realm specifically, what we find is that we are often not only looking at comparisons with normative population data, but more effectively for a number of our measures, we're looking at changes in raw score and growth scale scores over time. And these are things that often, when utilizing paper, require multiple steps to be able to get to. When we use an ECOA process, all of this can be built in as a way of making it much more effective for being able to have the scores that are necessary and to check those scores as required. Next slide, please. ECOA is best able to support the use of perfos and observers specifically because it is developed to provide built-in guidance for the raters. Utilizing tablet administration means embedding step-by-step prompts for these complex scales that we might be using, as well as streamlining administration and improving scoring accuracy, areas where we are often having to do many different checks when we're looking at paper-obtained data specifically. As a result, the validity of the data is stronger. These real-time checks that can take place during the administration of the scale as well as post-scale administration, and the ability to embed guidance that is available so that someone who is using a scale that they're very familiar with, they've been well trained on how to use it, but also may require time to time some checks in terms of making sure that, oh, for this population, for this age, this is where I need to start. This is what I need to be thinking about. These are the kind of areas of data that I need to be paying most attention to in terms of administering the scale. It allows for then standardizing the approach in which it's done and ensuring that then scoring that takes place is accurate. One of the key demands, particularly with performance outcome measures, like the Bayley scales of infant and toddler development or the Wechsler scales, are that there are basal and ceiling requirements that are often important to have to be keeping track of, but that also need to be addressed in different ways given the age levels of the population and their ability level. And this can all be built in terms of appropriate ECOA considerations and administration considerations that can then be reminded to the rater while doing the evaluation, but also can be utilized for doing independent review that takes place. And one of the things that the regulating organizations have basically been really aiming for is the ability to have the most reliable and the most objective data. And so it is often the case now that we are required to be able to provide traceable review processes to give feedback and actually remediate with regard to administration and data errors in a way to ensure that raters continue to be reliable and valid in their administrations. Utilizing ECOA improves the administration quality so that we can deliver reliable data for analysis. Next slide, please. A key area that we found that is actually both a challenge and also an area where there's a very ready solution is in the acceptability of ECOA tablet use as part of working with complex and challenging pediatric populations in particular. We've found that for many raters who are uncomfortable with the move to ECOA for their administration of their performance outcome measures specifically, They are concerned because they're used to utilizing a paper approach when assessing patients, and they're worried that by moving to a system that may feel less straightforward, because even with training and experience, it is still something that they may not be as automatic with, that it might interfere. Another challenge that comes up is also the fact that for many rare disease populations and neurodevelopmental populations, tablets are actually a daily part of these patients' lives. They are used for engagement, for teaching, and for entertainment. And as a result, there's a worry that because these individuals are very interested in tablets, that it may make it very tough for them to be able to be responsive to the testing as it takes place. To address this, what we've come to understand is that it really comes down to how to build in an effective training process. And this is something that can be done not only in preparing for the clinical trial and building it into the protocol as a process for getting the site raters skilled and confident in their work with the tablet. But it's also the case that it can be utilized from the baseline intervention kind of tool as a baseline intervention tool, where we actually help the patients and the caregivers become comfortable with the fact that a tablet is being used and being brought into the setting. And so ultimately what we found is that there are ways of actually guiding sites and the raters at those sites to become very expert at the use of the tablet and to actually take advantage of what the tablet provides, which is very much different than some of what happens with paper, where you're having to kind of go back and forth across the protocols themselves. Next slide, please. I want to talk about now an example of where utilizing eCOA proved to be a very effective opportunity for ensuring that a Gaucher disease clinical trial could move forward effectively, even with some of the challenges that automatically came with the fact that we were working in a pediatric study with a Gaucher disease type 2 presentation that was fairly severe in the patients who were participating in the study. The major sponsor who was investigating a new compound led them to develop a protocol that very quickly became clear would benefit from the use of eCOA for the administration of the measures that they were interested in taking advantage of. One that could actually make it much more, I would say, probably efficient during the administration at the site, as well as to be able to quickly have data that could be examined by the sponsor team in blinded manner as a way of ensuring that they were obtaining valid and reliable information about the drug and its effects. The goal with this clinical trial was to assess the drug's capacity to improve both cognitive and motor development, as well as to see if it helped decrease the pace of neurodevelopmental impact in GD2. This was a study utilizing a number of key scales across performance, observer, and clinician report, all administered with this study with our assistants through use of a tablet. Scales that were used included the Bayley scale, the Vineland Adaptive Behavior Scale, the GMFM88, a motor measure, and the CGI severity and improvement scores that were necessary for getting a sense of how the clinician was viewing change. These were actually utilized as both key secondary and even primary measures for this indication. Next slide, please. The study team was trained to utilize the tablet to administer the scale. This was a multi-site study. This was across a number of different countries. And so we had a varying level of experience with these scales. Most people were pretty expert, but there were sites that had raters who actually were still more novice and needed some time to become more proficient with that as well. So the training was a key element, and utilizing the tablet actually was an effective means for improving skill and also having ready guidance available for these raters, particularly the ones who were more junior in their administration, so that they could actually be more reliable. It also allowed us as the team working with them to monitor the success with which the training had been taken and in terms of how they were mastering the use of the scale, so that we had an ability to look at reliability across sites. The regulators had also requested a number of important considerations with this study because of their concern about the need for an objective evaluation with regard to the report that the VABS requires from the caregiver. We had independent rating. We had clinicians who were highly skilled with the VABS being able to administer these to the caregivers, done through a virtual process that was managed through the tablet. Likewise, we had a group of clinical scientists who were involved in performing independent review. So we had to video record at the sites the administration of both the Bailey scale and the GMFM-ADA and ensure the quality of those video recordings, as well as to serve as checks for the administration scores and their reliability. The study team was provided as a result with feedback from both the independent review of the BSID and the GMFM to ensure that there was reliable and standardized scale administration, as well as appropriate scoring taking place. And by using the tablet form for the administration of the study, administration and accompanying potential scoring errors were able to be identified quickly and remediated in order to ensure that we actually had effective data. Next slide, please. In evaluating how the study proceeded, we actually were able to play a very strong role because of both the use of ECOA and IREV. The study unfortunately had to be discontinued early as a result of several challenges, including some deaths of patients who were highly impacted by the disease process. The data that was obtained for all subjects, though, was able to be reviewed that in a way that provided a consistent process of both examining score administration, the administration of the evaluations and their scoring, so that we could actually then make good use of the independent reviewer scores as a guide for determining where intervention might be needed, as well as also varying checks along the way. The regulatory team was actually very pleased with the fact that this was built into the process in a way that allowed them to see that the independent review data was efficacious, and they actually chose to utilize that as a key index of how the study was proceeding and also the validity of the study and the drug's success. We held a number of lessons learned sessions throughout the process as well, between ourselves with Clario and the sponsor, in terms of identifying and intervening with site challenges in scale administration and developing an effective remediation protocol that was able to then ensure that we had reliable data across all sites. Additionally, the statisticians for the sponsor were able to actually feel very assured that they were meeting the regulatory considerations, as well as their being able to answer the questions with regard to the data's efficacy. And I think that this may be my last slide. So I'm going to now pass it over to Dr. Patel. Thank you.
Thank you very much, Dr. Hunter. Again, these all have been very excellent presentations and on point as to what to consider in rare disease. And I'm going to talk about the next 10 minutes about what cardiac solutions assessments you need to consider for a rare disease indication. So we can go to the next slide, please. So there are really three main assessments you should be considering for rare disease, and this all depends on if the disease contains or induces cardiac involvement or not. So the 12-lead ECG is the mainstay of assessing cardiac safety for every clinical trial, be it a rare disease or a common disease indication. And for every rare disease indication, each participant should get at least a screening ECG to assess the extent of any underlying cardiac disease. If the disease process involves myocardial involvement, then there should be on-treatment ECGs scheduled at appropriate intervals to follow disease progression and monitor safety. For patch and Holter monitoring for rare arrhythmic events or conduction block, again, if cardiac involvement is a feature of the disease indication, then these should be written into the safety monitoring plan to, again, protect patient safety and assess any kind of efficacy endpoints, as we'll see for certain rare diseases. And then finally, cardiac imaging is also indicated in rare disease indications where there is myocardial involvement. Doppler echocardiography is often preferred over cardiac MRI, particularly when there's valve involvement or pulmonary hypertension, because the Doppler can assess these gradients better than cardiac MRI can. It's also better accessible in remote areas and more third world countries where some of these patients may be more prevalent. We're talking about Gaucher disease type 1 as a model disease, and this is a classical lysosomal storage disease with significant cardiac involvement. And so I'll follow this trend as we go through the next few slides of what you get to assessments you get for cardiac safety and efficacy in Gaucher disease. You can go to the next slide, please. So ECG monitoring is critical in Gaucher type 1 disease. These patients have significant myocardial infiltration with the GB3. It induces myocyte-myocyte disconnection. Also, there's infiltration of the fibroblasts in the heart, which can also lead to zones of slow conduction and arrhythmogenic foci. For this reason, you need ECGs to assess AV conduction blocks, conduction disorders. They're also prone to pre-excitation syndrome, and the 12-lead ECG is perfect for capturing these at screening and following progression throughout the trial. You want to consider getting ECGs at screening and again at appropriate time points post-dose, as well as end of trial, to adequately monitor patient safety. The other consideration here is to get centralization of ECG collection and overreads to reduce variability. Again, with small patient populations, variability is going to be quite high. And so anything you can do to reduce the variability to improve the precision of the assessments will help with these small cohorts. The centralization reduces variability from site to site and different ECG machines. This leads to fewer false positive findings and fewer false negative findings to protect patient safety. The ECG shown here is a typical one from a screening ECG of a patient with type 1 Gaucher disease in a clinical trial. It shows low voltage and a left bundle button shock, consistent with significant myocardial infiltrative disease. We can go to the next slide, please. Continuous ECG monitoring is also warranted in type 1 Gaucher disease. Again, because of the significant myocardial infiltration, there's a propensity for more atrial arrhythmias, ventricular arrhythmias, and conduction block. These are rarer events that a 12-lead ECG won't often capture on just 10-second duration of recording. And so usually you're going to use a patch monitor for safety assessments to assess the burden of these arrhythmias and conduction blocks in type 1 Gaucher disease. However, if you're looking at certain rare diseases where PVC burdens and NVC counts can be primary endpoints, this is more in genetically induced cardiomyopathy, such as arrhythmogenic right ventricular dysplasia or catecholaminergic polymorphic VT, then Holter assessment of multi-leads might be a better assessment than the patch. And this is shown in the figure in the right-hand lower corner, where the 3 leads in panel A are from a Holter monitor. The ectopic beat towards the middle of the trace, it clearly shows that it's a wider complex, more consistent probably with a ventricular ectopic beat. Whereas if you look at panel B in the bottom, which is a patch recording with a single lead recorded simultaneously in this patient, that narrow complex ectopic is unclear if it's ventricular atrial in origin. So Holter monitoring conducted for 1 or 2 days at appropriate time points during treatment can be better for efficacy assessments, whereas patch is good for safety assessments. The important thing with both of these and continuous ECG monitoring is to remember that you're collecting large amounts of data. And it is important to collect appropriate amounts of pre-dose and appropriate placebo dose data to make sure that any findings that you're seeing can be related to either being pre-existing to the disease or related to the treatment. And so if you don't have adequate pre-dose assessment or control doses, this becomes a very difficult issue to resolve. And that's something to very important to consider when you design your study for continuous ECG monitoring. Go to the next slide, please. Cardiac imaging is also warranted in Gaucher type 1 disease. These patients are prone to ventricular hypertrophy, ventricular dysfunction. They get aortic stenosis and pulmonary hypertension, which is due to the hepatic buildup of GB3 in the liver, inducing portal hypertension, then that reflects up through the right ventricle into the pulmonary circuit. As we discussed before, Doppler echocardiography is superior actually to cardiac MRI in this situation for assessing valve stenosis and pulmonary hypertension. It's also more accessible, and so it's easier to use where rare diseases might be in more remote locations that you're recruiting. Again, it's important, like with ECGs, to get an assessment at screening, pre-dose, to determine the extent of myocardial involvement the patient has, and they get appropriate on-treatment echocardiograms at appropriate timed intervals to check disease progression and monitor safety. And again, like with ECGs, centralizing ECG collection and assessment can minimize and reduce variability to reduce false positive safety findings and false negative findings to both protect the patient safety database and protect patient safety. The image here is again from a patient with type 1 Gaucher disease. The image on the left shows a 2D dimensional echocardiogram. There's a slightly thickened aortic valve with aortic stenosis, which is typical of the disease. And the right ventricle at the top of the image is thickened and enlarged, indicative of longstanding pulmonary hypertension. The image on the right is a longitudinal strange heat map, and again, consistent with an infiltrative disease process where there's heterogeneous changes in longitudinal strain. And we can go to the next slide. One thing I really want to emphasize with rare diseases is that a thorough QT and QT assessment is still required, despite the fact that you have small cohorts. And there's been a lot of confusion around this when we discuss these kind of programs with clients and sponsors. So if the therapeutic is a small molecule asset, it does have a high risk of off-target potential repolarization effects. And you do need to get a QT assessment to assess the extent by which QT prolongation may occur. With rare diseases, you're obviously going to have small dose cohorts. And so the regulators are fully understanding of this and will accept underpowered studies, but they will not allow you to forego a QT assessment or give a higher risk margin. You still have to meet the 10 millisecond threshold with a rare disease, not a 20 millisecond threshold as sometimes is allowed for oncologic therapies. If the asset can be safely to healthy volunteers, the expectation may be that you do a more thoroughly powered assessment of healthy volunteers at supertherapeutic doses to really get good data to rule out a QT prolongation effect. If the asset cannot be given safely to healthy volunteers, then you can trigger ICHE14 6.1 pathway, where you can conduct a thorough QT assessment in patients at the clinical exposure. You don't have to go to supertherapeutic exposures without placebo or positive control data. But again, the expectation is that you can rule out a 10 millisecond effect, not having a higher margin of 20 milliseconds just because it's a rare disease. Like with all indications, you don't have to do a thorough dense PK ECG assessment for large biologic molecules, such as a monoclonal antibody. Just collecting appropriate safety ECGs throughout all phases of development will be adequate. But the FDA has required more thorough QT assessments for RNA therapeutics, particularly those that might have propensity to accumulate in the myocardial tissues, or biologically engineered proteins that may have potential off-target effects. And again, this is something that will be discussed on a case-by-case basis. We can go to the next slide, which I think is the last slide I have, and just summarize what I've gone over here for cardiac assessments. So all participants in every rare disease indication should get a screening ECG and an end of treatment ECG. If the disease involves the heart with myocardial dysfunction, then appropriate on-treatment ECGs should be collected regularly to monitor safety. Continuous ECG monitoring with a patch is appropriate for safety monitoring in diseases with myocardial involvement. Holter may want to consider if you have efficacy endpoints. for PVCs or non-sustained arrhythmias. Doppler echocardiography is the preferred method for imaging if the disease involves valvular dysfunction or pulmonary hypertension. It's preferred over cardiac eye because of easier accessibility. And finally, there is a requirement to do a third QT assessment for all small molecule therapies and some nucleotide therapies and engineered proteins. The regulators will accept underpowered studies, but not a higher risk QT assessment. And with that, I'll hand it back to Dr. Mark Oppler to conclude the presentation. Thank you.
Dr. Patel, thank you so much. Before we go on, let me just summarize what we the panel have sort of put together as the key takeaways for today. First, obviously, rare disease trials have unique challenges. From the small cohorts to the subjective endpoints, there's a demand, I think, as we've seen in today's discussion, for adaptable, flexible methods. Next, as noted by Dr. First, imaging endpoints have to be pragmatic. Engaging regulators early, right-sizing what's being measured, and centralizing both QC and analysis, as well as prioritizing safety, are going to be critical to success. Third, as we heard from Dr. Hunter, ECOA done right strengthens clinical endpoints. Built-in tablet guidance and real-time checks, as well as techniques like independent review, can improve administration, scoring, and data reliability. And last but not least, from Dr. Patel, the critical importance of cardiac safety in all of our work, obtaining universal pre-dose and on-treatment ECGs, adding Holter patch and Doppler echo when indicated, and assessing QTC per modality specific expectations is vital. If we go to the next slide, I think we're gonna wrap that up and move on to questions. Thank you to those of you who submitted questions, and I want to invite the audience to continue to submit questions as we have a little bit of time left to cover these. First questions that's come in, Thomas, I think this one is for you. And the question is there a radiation limit for clinical trial research, and does it vary between adult and pediatric participants? And as I am definitely not qualified to answer, I will turn that over to Dr. First.
Yeah, thank you, Mark. Yeah, this is a common question that we get. Unfortunately, there is no established universally accepted radiation limit for exposure of research subjects. But that limit is really going to be context dependent based on how much imaging is done in standard of care and how the trial imaging relates to that, and also life expectancy, since the primary concern is carcinogenesis, which can take 20 years or longer to materialize. There are, however, US and international radiation limits for the class of radiation workers, and that limit is at 50 millisieverts per year, which is about 15 times the amount of radiation we get from the environment, naturally occurring radiation. So it's a relatively high limit that covers really most indications, perhaps with the exception of oncology, where there might be frequent use of CT or PET CT scans. Turning to the pediatric population, there has been some discussion that a threshold that's 10 times lower than the adult threshold may be appropriate for pediatrics, just because of the sensitivity of that population and the long life expectancy. But again, it's context dependent, and we have seen situations where that limit was higher in certain indications.
Thank you very much, Thomas. Much appreciated. Scott, I believe this next question is for you, and it says, how can we get ECOA to be more effectively accepted by site raters who work clinically and are more familiar with paper-based forms?
I think that's an excellent question. And as I was discussing with the presentation, it's one of the key considerations that we keep in mind whenever we have a new study coming forth, particularly in the rare disease space, because again, these are complicated scales and also test batteries that are utilized often. And the raters at sites tend to be very well-skilled individuals, but because they are predominantly clinicians, they are often utilizing the tests as they are established with regard to paper administration procedures. And as a result, what we've found is the best way to actually prepare for this is to determine the level of opportunity that's necessary for spending time with the tablet, learning how to administer and take advantage of the key elements that are able to be built into this format, so that then it becomes a much smoother process and one that they feel more secure with. It's also the case that we want to have them have the opportunity to kind of practice as much as possible too, so that they are able to then, while working with the patients, feel that the tablet is an extension of them.
Practice makes perfect.
Very much so.
Thank you for that. Very, very helpful. Vic, I think this next question is for you. It says here, you mentioned that echocardiograms are better than cardiac MRI to assess pulmonary hypertension and valve function. But how does it compare for measuring LV mass?
Thanks. That's a very good question. And an important aspect that you're really going to want to assess all of these features for most rare diseases. So where you're really going to have the benefit with the Doppler echo for measuring gradients, when that's a feature of the disease, of the valves or pulmonary hypertension, you can still collect LV mass assessments with echocardiography. It's not as precise as cardiac MRI, but it's usually good enough. And I think the issue is, of course, if you have to do the valve assessments on pulmonary hypertension, you're really not going to get that with cardiac MRI. And so you don't have to do both assessments. You can still get LV mass and function with echo well enough with modern, even 3D imaging modalities now are available or have a better precision than 2D. So it's poorer than cardiac MRI, but still quite good enough to meet the needs for most studies.
Thank you for that. Much appreciated. Thomas, I think we have just enough time for one more question, and I'm going to inflict it on you, if that's okay. Question that's come in, it says here, with increasing competition for patients in rare disease indications, what are some strategies to finding study participants? And, I'm pitching it to you, Thomas, but I think I'll also open it up to the rest of our colleagues here if they want to chime in. But feel free to lead us off if you'd like.
Yeah, thanks for the easy softball question. Yeah, so obviously, as discussed, expanding into new geographic regions to find patients is a strategy, but those are getting rarer and rarer as the field has looked for these patients. One strategy that we've seen used is to initiate early in the trial development, or the drug development phase, a natural history study in which patients are enrolled and recruited into a trial, not given any intervention, just followed over time. And this has a couple of benefits. Number one, it adds to our knowledge of the disease progression, and we gain more information about how patients proceed with their disease. But it also establishes a relationship between investigators and patients, and identifies or creates your own database of patients that when your phase 2 and phase 3 trials are ready to enroll, you can tap into that patient population and move them into a clinical trial setting after having established that relationship. And we've seen this done in many different indications, and it seems to be a very effective way to jumpstart enrollment once you've reached phase 2 and phase 3.
Thank you for that. I think we're probably out of time, although we have a great many more questions. But I'm sure I and my colleagues will be happy to try to address them offline. Thank you, everybody, again for joining us today. Ryan, I'll pass it back to you to wrap up.
Thank you all very much for today's very insightful presentation and discussion, but we have reached the end of our time today. Now, if we couldn't attend to your questions, know that the team at Clario will follow up with you, or if you have some further questions, you can direct them to the contact info that's up on the screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Additionally, I've shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. For those watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and then gain access to features such as viewing the recording and more. Now, please join me once more in thanking all of our speakers for their wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thank you for coming.
Thanks, Howard.
Thank you.
Thank you.